This video is part 2 of membrane structure and molecular transport, mainly dealing with the transport of molecules across the cell membrane. Again, here's a picture of the cell membrane that you'll probably become very familiar with. A couple of just introductory matters. Membrane transport, a transport across the membrane can happen in many ways. It can just happen with something simply just being able to diffuse across the membrane, much like this. However, a lot of times this happens with the help of a transport protein like that looks kind of like a tube. It could happen with one of these grabber type proteins that goes and literally grabs the solute out of the solution. And so it could happen a number of ways, but in general, the way that transport happens is through a process called diffusion. And diffusion is the transport of materials from a high concentration to a low concentration. Those materials can be salt, sugar, whatever, orange dots, purple dots, anything, or they could be water. Water could be the material that's being transported. No matter what, it goes from high concentration to low concentration because there is something that's built up called a concentration gradient. This represents a gradient. There's high here, low here. It's like your roof or a hill in the street. There's a high place and a low place. If you want to roll down the hill, say on your bicycle, does it require any energy? No, it actually makes energy. You could actually use that energy to do something. So in much the same way, diffusion is a form of what we will call passive transport, which is movement or movement of materials without the need for energy. And in many cases, like we said, it will create energy. So you can see these simple diagrams here. Materials are going to move to the right until everything's equal. When everything is equal, that state is said to be equilibrium. Equilibrium is not where everything is still. Things are still moving around, but they're moving at an equal pace. There are as many going right as there are going left. It's very similar to the bottom, except for this time you have two solutes moving back and forth. Same kind of idea. Now, there are types of diffusion. The kind that we're going to spend a lot of our time is the diffusion of water, which is called osmosis. So how does this work? Well, we look at this particular setup here. And let's say that this, well, it says here, this is a selectively permeable membrane. In this case, it does not allow the passage of purple dots, but water can pass through. So, just speaking of purple dots alone, purple dots here are high, here they're low. Could they move? They would move from right to left, but they can't move. Water, on the other hand, which side is high? Well, there is more water over here because there's less purple. There's less water over here. So which way does the water want to move? From high to low. When we are dealing with a system where water is going to be moving, we're going to use terms hypotonic and hypertonic. These two terms are comparative terms, meaning you never use them alone, and they are also in reference only to the solute. Hypo means less, hyper means more. So the right side is the hypertonic solution because there is more solute than on the left. When we're dealing in this kind of situation, water always moves from hypo to hyper, hypo to hyper every time. And so what will be the resultant of this situation? The water will move until the water itself is at equilibrium. This happens with living cells, with living systems, quite a bit, obviously. Here we put this a normal animal cell into a hypotonic solution. So water, there's more water on the outside than there is on the inside. And so water again moves from hypo to hyper, causing that plant cell to, or that animal cell to break. The plant cell, on the other hand, water's moving in, but it's not going to break because that plant cell has a cell wall. And so plants that have this sort of condition are said to be turgid, which is a normal condition for plants. In an isotonic solution, what do you think isotonic means? Well, it just means when the water is equal on both sides. In an isotonic solution, water is equally moving in and out. Animal cell, normal. Plant cell, this is when the plant cell is going to begin to look wilted or flaccid. If you put that same cell in a hypertonic solution, where the inside of the cell is hypotonic and the outside is hypertonic, water is going to move out and it's going to cause the cell to shrivel. Or in a plant cell's case, it's going to cause it to plasmolyze, or the cell membrane will actually separate from the cell wall. Now, you said earlier that the movement of water, or the movement of materials can be used to power things in the cell. I mean, think about the way that we use dams to create electricity. Very similar. This concept is called water potential. Water's ability, when water's the thing moving, water's ability to perform work while passing through the cell membrane. Water always flows from, and this is the symbol for total water potential, always flows from a high total water potential to a low total water potential. Well, how do we figure total water potential? We're going to add the pressure potential. Think about it. If you put pressure on water, negative or positive pressure, positive would be like pushing down, negative would be like pulling, like a plunger in a toilet. That's going to cause the water to move. Well, what else can cause water to move that we've already looked at? Solutes, right? And so a solute is also going to have potential to cause the water to move. And so you add these two things together, you get the total water potential. And usually we're going to be comparing one system's potential to another system's potential. So that's what we see here. On the left, you have, fill in the blanks here, there's no pressure because there's nothing acting on that side. There's no solute in the water either. No solute. And so 0 plus 0, 0 total water potential. On the right hand side, there's a positive pressure pushing down on this system of 0.3. It's an arbitrary number. The pressure potential will always be given to you. And the solute potential is actually it will always be negative, because what is it trying to do? It's actually always pulling the water towards it. It's never pushing the water away. And in this case, the solute potential is negative 0.23 with a total water potential of 0.07. Water moves from high to low, so it's going to be moving to the right in this situation, or the left, excuse me, in this situation. Here's another one where negative pressure is the water, you can see here, is being pulled. Negative pressure, but no solute. You get the idea. The water's wanting to go this way because of the pressure. It's wanting to go that way because of the solute. Which one's higher? Well, negative 0.23. One of the mistakes that most of my students make, even though some of them are in classes like calculus, they will look at 0.23 and 0.3 and decide that 0.3 is higher, but they forget that their number line, negative 0.23 is higher, so this will go to the left. I said you'll never have to solve for pressure potential, but you will have to solve for solute potential. Here is that equation. So a couple things here. Notice I did say that the solute potential will always be negative. For some, they would rather see this equation written like this. And I'm okay with that. Just know that that negative is going to cause the whole thing to be negative every time. So let's talk about these different numbers in the context of our class. Concentration, you're usually not going to have the figure for this. It'll be given. It'll be a number in moles, and it's always given. I like the pressure. The R, the pressure constant, is the same every single time, 10831. Temperature in Kelvin, notice. So you will usually be given the temperature in Celsius. So how do you get Kelvin? Celsius plus 273 equals Kelvin. So if the temperature is 27 degrees Celsius, it is 300 degrees Kelvin. Why is that? Why Kelvin? Well, science uses Kelvin because there's no negative. Zero is the absolute number. Literally, it's absolute zero. And so really the only hard portion of this problem is the ionization constant. So let's talk about the ionization constant. The ionization constant will usually be 1 or 2, and it has to do with the number of pieces, the number of parts that the particular chemical will ionize into. So take something like sodium chloride. We know that sodium is a positive ion. Chlorine ionizes by gaining an electron, so it becomes negative. If you put sodium chloride in water, it breaks up, right, into two pieces, sodium and chlorine. And so its ionization constant will be 2. The same will happen with acids and bases. So let's take a base like sodium hydroxide. We know that hydroxide is a negative ion. Again, sodium positive, so the ionization constant will be 2. You could do the same thing with hydrochloric acid or any other kind of acid or base or salt. With sugars like glucose or sucrose or other, the ionization constant is going to be 1. Remember, sugar is bound up with covalent bonds, it does not ionize in water, and so it does dissolve in water because it's polar, but the ionization constant of sugars will be 1. And so that number ionization constant is either going to be 1 or 2 based on the type of chemical that it is. Find out all those numbers, multiply them together, you get your solute potential. Not that difficult. Here are some problems. I'll just do a close-up on these so you can look at them yourself, but I'm not going to go over every one of them. Another type of diffusion that is passive transport as well is called facilitated diffusion. Facilitated diffusion is very simple in that it is the transport of materials with the help of a transport protein. That is it. A protein facilitates the diffusion of materials. A lot of times that's what we're going to be looking at in this class. And so switching gears to active transport. Active transport is a process that requires energy, just like pushing a car uphill, pushing things that are trying to fall out of the closet into the closet and so forth. It requires energy. You're storing up a gradient in order to do work. It's creating a gradient, like blowing up a balloon. Once you blow up the balloon, what can you do with it? You can let all the air out and blow out. It'll create energy. There are lots of examples of this in the cell. Here's an example that we will talk about in a later unit. This is a sodium-potassium pump. Think about the word. It's a pump. If you take your bicycle tire pump, you don't just look at it, and you have to actually physically go pump air into your tire. And each time you see the word pump, it is something that requires energy. This is an example of voltage gradient that is in nerves. It also is created, well, it is created with those sodium potassium pumps. It is something that is set up to do work. Proton pumps, we'll talk about those when we talk about photosynthesis and cellular respiration. Pumping protons to create a gradient in order for that gradient to create ATP. So there are several examples of this sort of thing in cellular processes. Another type of active transport is called endocytosis. Endocytosis. Two types of endocytosis, phagocytosis and pinocytosis. Both essentially the same idea. They are taking a large particle, food with phago, meaning cell eating, pino, cell drinking, liquid, so that's liquid, and taking a large particle in, creating this little vacuole out of the cell membrane itself, kind of pinching off, and then that material will be used by the cell somehow, probably combined with a lysosome and broken down further. The opposite of this is called, well, I don't have a picture for this. Here's another type of endocytosis that just requires a receptor, receptor-mediated endocytosis. The opposite of this is called exocytosis, in which you have a cell, there's some sort of vacuole of food, or maybe let's say this vacuole's got some proteins that the cell is attempting to ship out. All right, and this vacuole will move to the surface of the membrane and will literally expel those materials right out into the space. That is exocytosis. Again, endocytosis and exocytosis are both forms of cellular transport that require energy.